Hello, I am Dr. Owen On, and welcome to the next installment of the Boring Lecture Series. In this lecture, we will be talking about annual milk yields of goats. This lecture presents a comprehensive examination of factors affecting annual milk yield of different goat breeds using panel data analysis. The study in question was conducted using data from 5 different goat breeds raised in Turkey during the period spanning from the year 1991 through the year 1997. The breeds examined in this investigation included Damascus hybrid goats, pure Damascus goats, Tauros goats, Cukurova goats, and German fawn goats. These breeds were selected to represent both local indigenous breeds and culture breeds that have been introduced to the region over time. The data collected for this study included annual milk yield measured in liters, which served as the dependent variable in the analysis. Additionally, the lactation period measured in days and the age of the animals measured in years were recorded as independent variables. The study employed panel data analysis methodology, which is an econometric method that provides advantages in analyzing agricultural production data due to its capacity to account for both temporal variation and cross-sectional variation simultaneously. Goat farming represents an important source of livestock production in terms of milk production, meat production, and fiber production for human populations throughout the world. Goats are known as one of the most preferred animals for husbandry purposes due to their high adaptability to varying environmental conditions, their low maintenance requirements, and their demonstrated ability to adapt to different geographical regions across diverse climatic zones. In terms of these features and characteristics, goat farming makes an important and substantial contribution to sustainable agricultural practices in many regions of the world. Dairy goat farming specifically is an important source of income, particularly in the context of small animal farms where resources may be limited and efficiency is paramount. However, there exist many biological and environmental factors that affect milk yield in dairy goats. The most important of these factors are breed differences, which reflect the genetic potential of different populations, the age of the animal, which correlates with physiological maturity and productive capacity, and the lactation period, which represents the duration of time during which the animal is actively producing milk. While the genetic potential of different goat breeds is a determinant factor of milk yield, physiological development and production capacity also change as a function of age. The lactation period is an important parameter affecting both the quantity of milk yield and the quality of milk produced. In addition to these primary factors, there are numerous secondary factors that may influence milk production, including nutritional status, health status, environmental temperature, housing conditions, management practices, and various other variables. The panel data analysis method is an important econometric method that is preferred in the analysis of agricultural production data because it provides more observations and greater statistical density by taking into account both the time dimension and the unit dimension of the data structure. Since this methodological approach offers the opportunity to control for heterogeneity between variables, it allows for more accurate estimation of the effects on production performance of different goat breeds. In the analyses that were performed in this study, fixed effects models and random effects models were compared using statistical testing procedures. The appropriate model for the data structure was examined and selected with a Hausman test, which is a statistical test designed to discriminate between these two model specifications. The panel data structure that was employed in this research revealed the effects of factors that change over time and factors that differ between breeds in substantially more detail than would have been possible using conventional cross-sectional or time series analysis methods. The findings that were obtained from this investigation are intended to provide a scientific basis for practical applications such as breed selection and age management selection in order to increase productivity in goat breeding operations. This study aims to comparatively evaluate the performance of the 5 different goat breeds in terms of milk yield, lactation period, and age using panel data analysis methods. The goal is to achieve sustainable productivity increases in goat farming through the analysis of biological factors affecting production performance using rigorous scientific methods. Accurately identifying the variability in milk yield among different breeds plays a critical role in both guiding the breed preferences of producers and in regional and interregional production planning activities. In this context, Evaluating long-term datasets containing multiple breeds using econometric methods offers the opportunity to more objectively examine the effects of both genetic potential and physiological traits on production outcomes. The study was conducted with data that included milk yield information, lactation period information, and age information of 5 different goat breeds that were raised in Turkey during the period between 1991 and 1997. The goat breeds that were examined in this research were Damascus Hybrid, Pure Damascus, Toros, Cukurova, and German Fawn breeds. These breeds were selected deliberately to represent both local indigenous breeds and introduced culture breeds. The annual milk yield, which was measured in liters, the lactation period, which was measured in days, and the age of individual animals, which was measured in years, for randomly selected individual goats from each breed were recorded on a regular systematic basis between the years of 1991 and 1997 inclusive. The data set that was used in the study included the following variables on an annual basis for the 5 different goat breeds during the period between 1991 and 1997. The dependent variable was annual milk yield measured in liters. The independent variables included age measured in years and lactation length measured in days. The breed designation served as the panel cross-section unit identifier. The year, ranging from 1991 to 1997, served as the time dimension of the panel structure. The research model is based upon panel data analysis methodology, which has both cross-sectional dimensions, represented by the different breeds, and time dimensions, represented by the years from 1991 to 1997, which allows for the analysis of how variables differ over the course of time. The panel data method produces results that are more accurate and more consistent when applied to agricultural and animal production data, owing to the ability to control for fixed differences between individual subjects or groups. In the study that is being described in this lecture, Pearson correlation coefficients between the determined characteristics were calculated first in the analytical sequence. Variables were standardized by regression analysis procedures, with actual milk yield serving as the dependent variable and other measured variables serving as the independent variables. Panel data analysis was applied in the analysis of the data using the statistical software package of use version 9. In the study, the first step in the analytical process was to perform Spearman correlation analysis to determine whether there exists a multicollinearity problem among the independent variables that were included in the model. Multicollinearity refers to the situation in which there exists a complete or nearly complete linear relationship between all or some of the independent variables in a panel regression model. A high degree of correlation between independent variables can make it impossible or extremely difficult to calculate the parameter estimates and can render the least squares estimation method unusable or unreliable. Therefore, in panel data analysis, it is necessary to first investigate whether there is a multicollinearity problem among the independent variables. This investigation was carried out using Spearman correlation analysis and variance inflation testing, which are both frequently used methods in the statistical literature. Variables that exhibited values above the critical threshold values that may cause a multicollinearity problem were identified for potential removal from the analysis. Subsequently, the dependency between the cross-sections that form the panel was examined using the Lagrange multiplier test and the cross-section dependence test on the basis of the panel as a whole and on the basis of individual variables. If there exists cross-sectional dependency between the series, this situation should be taken into consideration and the analysis should be carried out accordingly, as this condition affects the accuracy and reliability of the findings that are obtained. Analysis results that do not properly account for cross-section dependency may become biased and inconsistent. In this direction, it is necessary to test whether there is cross-sectional dependency in the series before proceeding with panel data analysis. The existence of cross-sectional dependency between the series can be determined by the Lagrange multiplier test, the cross-section dependence test and related variance, or the adjusted Lagrange multiplier test. The Lagrange multiplier test is used in circumstances when the time dimension is much larger than the cross-section dimension. The cross-section dependence test variant is used when the time dimension is larger than the cross-sectional dimension, but the difference between the two dimensions is not particularly large. A different version of the cross-section dependence test is used when the cross-section dimension is larger than the time dimension. The adjusted Lagrange multiplier test eliminates certain statistical deviations that can occur in the standard Lagrange multiplier test and addresses the possibility of the total correlation in the Pesaran cross-section dependence test being equal to 0. And this adjusted test is used when the time dimension is larger than the cross-section dimension. The existence of cross-sectional dependency among the units forming the panel was examined separately on a panel basis and on a variable basis. The variability of the coefficients of the variables between the cross-sections was investigated using the homogeneity test. The homogeneity test was examined separately according to whether the constant terms and the slope coefficients of each variable were homogeneous or heterogeneous on a variable-by-variable basis, and also when excluding certain explanatory variables from consideration. In order to perform panel data analysis and obtain accurate and meaningful results, the stationarity of the time series of the variables must be ensured. In other words, in order to obtain meaningful and interpretable results representing the relationships between dependent and independent variables, the series must be stationary. In panel data analysis, it is necessary to determine whether the cross-sections are independent of each other before conducting the unit root test for stationarity. At this juncture, panel unit root tests are divided into two groups, which are designated as first-generation tests and second-generation tests, based on their treatment of cross-sectional dependency. First-generation unit root tests are based on the assumption that cross-sectional units are independent of each other, and that a shock occurring in one of the sections forming the panel affects all sections at the same level with uniform impact. In addition, these first-generation tests are divided into two groups, which are homogeneous models and heterogeneous models. While certain tests are based on the assumption of homogeneity across cross-sectional units, other tests are based on the assumption of heterogeneity across cross-sectional units. Some tests are based on both the assumption of homogeneity and the assumption of heterogeneity depending on the specific implementation. The second-generation unit root tests are based on the assumption that the cross-section units are not independent from each other, and that a shock occurring in one of the cross-sections forming the panel affects all cross-sections but at different levels of magnitude. In this context, the second-generation unit root tests were developed to properly account for cross-sectional dependence between the cross-sections in the panel structure. The stationarity of the variables that were used in the study was determined by taking into account both the cross-sectional dependency conditions and the homogeneity conditions that were identified in previous testing stages. As a result of the cross-sectional dependency test that was conducted, it was determined that cross-sectional dependency was detected between certain variables for the 5 different breeds under investigation. For the variables where cross-sectional dependence was identified, the Pesaran cross-sectionally augmented impesarantian test was used for stationarity testing. This test is a second-generation panel unit root test that accounts for cross-sectional dependence. The values of the test statistics that were obtained as a result of this analysis were compared with the critical values to test for stationarity for each variable. When the critical table value was greater than the test statistic value that was obtained from the analysis, the null hypothesis that the series has a unit root was rejected, and it was concluded that the series was stationary. For certain variables that were determined to have no cross-sectional dependence but to have a heterogeneous structure as a result of the homogeneity test, first-generation unit root tests that have a heterogeneous structure were applied. Before proceeding with panel regression analysis, the model must be tested for deviations from the assumptions of multiple regression analysis. The assumptions of multiple linear regression analysis include the absence of multicollinearity, the absence of heterosedasticity, and the absence of autocorrelation. Multicollinearity refers to high correlation among independent variables. Heterosedasticity refers to the situation in which the error term variance in the models is not the same for all observations. Autocorrelation refers to correlation between successive values of the error terms. Tests for these assumption violations were conducted and the results were examined separately for each panel data model specification that was considered. Fixed effects models and random effects models were estimated for the panel data. The Houseman test was applied to choose between the two model specifications. The Hausman test is a statistical test that examines whether the unique errors are correlated with the regressors in the model. The null hypothesis of the Hausman test is that the preferred model is the random effects model because the unique errors are not correlated with the regressors. The alternative hypothesis is that the fixed effects model is preferred. The Hausman test results indicated that the fixed effects model provided more reliable and consistent parameter estimates for the data structure being analyzed. Therefore, the fixed effects model was preferred and selected for use in the subsequent analyses. When the descriptive statistics results are evaluated, several patterns emerge from examination of the summary measures. The average value of milk yield for the German fawn breed is higher than the average values for the other breeds that were studied. Similarly, the average value of the age variable is higher for the German fawn breed than for the other breeds. Thus, the German fawn breed exhibits both the highest milk yield and the longest lactation period, which suggests that it may be an ideal breed for milk production purposes when those are the primary objectives of the breeding operation. In addition, it was observed that the average milk yield difference between the German fawn breed and the other breeds is substantial. This observation suggests that the superior performance depends on genetic differences between breeds and possibly also on differential adaptation to environmental conditions. When the skewness value is examined for the distribution of variables, it is observed that the skewness value is negative in all breeds and takes values that are relatively close to zero in magnitude. In other words, this indicates that the distribution of values is skewed to the left, though not extremely so. In the correlation matrix that was constructed to test the assumption of multi-collinearity, the correlation coefficients between the independent variables were examined. The problem of multi-collinearity manifests as a situation where there is a high degree of correlation between the independent variables. A correlation coefficient with an absolute value above 0.9 between pairs of variables is generally considered to create a problematic degree of multi-collinearity. When the correlation matrix was examined, the highest correlation coefficient between any pair of independent variables was that between the lactation period variable and the age variable, which was calculated to be 0.61. This value of 0.61 is not considered to be at a critical level in terms of multi-collinearity concerns. Thus, this finding indicates that there is no substantial multi-collinearity problem between the variables that were included in the model. Endogeneity is the situation where one of the independent variables in a regression analysis is related to or correlated with the error term. When the potential for endogeneity was examined, it was observed that the probability of a problematic relationship existing between the error term and the milk yield and age variables in the 5 different breeds was assessed to be low based on diagnostic testing. The existence of cross-sectional dependency among the units forming the panel was examined by performing panel analysis. The cross-sectional dependency test results revealed specific patterns. When the cross-sectional dependency tests that were performed on a panel-wide basis were examined, it was found that the probability value was greater than 0.05 in all tests that were applied for the annual milk yield variables of the Cukurova breed, the Damascus breed, and the Toros breed. In contrast, the probability value of all other variables was less than 0.05. Since the cross-sectional dimension was larger than the time dimension in this panel structure, the Pesaran cross-section dependence test results were taken into primary consideration for interpretation. As a result of the analysis, since the probability values of the annual milk yield variables belonging to the Cukurova, Damascus, and Toros breeds were greater than 0.05, which is accepted as the conventional critical value for statistical significance, the null hypothesis of no cross-sectional dependence could not be rejected for these variables. On the other hand, since the probability values of the other variables were less than 0.05, the null hypothesis of no cross-sectional dependence was rejected for those variables. While performing the stationarity test for the annual milk yield variables belonging to the Cukurova, Damascus, and Toros breeds, the conclusion was that there is no problematic cross-sectional dependence. Thus, while performing the stationarity test for these particular variables, first-generation unit root tests that do not account for cross-sectional dependence were deemed appropriate for application. If there is no cross-sectional dependence between the series in a panel data analysis, heterogeneity tests should be applied to determine which specific unit root tests should be applied for testing stationarity of the variables. The slope heterogeneity test was conducted to examine whether the slope coefficients differ across the cross-sectional units. The analysis results of the slope heterogeneity test were obtained for the 5 different races and for each variable separately. As a result of the heterogeneity analysis, it was determined that the probability values of the age variables for the 5 different breeds were less than the critical value of 0.05. Therefore, the null hypothesis of slope homogeneity was rejected for the age variable. In other words, it was determined that the slope coefficients for the age variable were homogeneous across the cross-sectional units. The probability values of the lactation period variable and the annual milk yield variable for the 5 different breeds were above the critical value of 0.05, and consequently the null hypothesis of slope homogeneity was not rejected for these variables. In other words, these variables exhibited heterogeneous slope coefficients across the cross-sectional units. Stationarity tests consisting of first-generation unit root tests that do not take into account cross-sectional dependence and that allow for a heterogeneous structure were applied for the lactation period and annual milk yield variables for the 5 different breeds. In addition, stationarity tests with a homogeneous structure were applied for the age variables for the 5 different breeds, given that homogeneity had been established for that variable. In the panel regression analysis, an attempt was made to determine whether there exists a statistically significant relationship between the dependent variable and the independent variables and, if such a relationship exists, to determine the direction and magnitude of this relationship. As a result of the analysis that was conducted, it was determined that the variances of the error terms in the model to be estimated were not constant across observations, which is the condition known as heterosedasticity. Furthermore, it was determined that the successive values of the error terms were not independent of each other, which is the condition known as autocorrelation. In order to address these violations of the classical regression assumptions, the period seemingly unrelated regression method, which is also known as the panel-corrected standard errors method, was employed to correct the panel standard errors. This method was developed to address the problems of heterosedasticity and autocorrelation in panel data settings. The estimation results that were obtained using this corrected method revealed important findings. The model estimated values showed that when the results are examined, the F-statistic probability values, which express the significance of the model as a whole, indicated that the regression models were statistically significant. The adjusted R-squared values, which indicate the proportion of variance in the dependent variable that is explained by the independent variables after adjusting for the number of predictors, were obtained with different magnitudes for the different breed-specific models. The adjusted R-squared value was calculated to be 0.45 for one model, 0.39 for another model, 0.64 for a third model, 0.55 for a fourth model, and 0.42 for the fifth model. These values indicate that the models explain between approximately 40% and 64% of the variation in milk yield, depending on the specific breed being modeled. In addition to the overall model fit statistics, the Honda test result was found to be statistically significant for the lactation yield of the Cukurova breed, the Damascus breed, the pure Damascus breed, and the Toros breed. The Honda test is a diagnostic test that examines whether there are structural differences across the panel groups. The statistical significance of the Honda test indicates that there is a structural difference in the lactation yield patterns across these different breeds. In other words, it can be stated with statistical confidence that there exist meaningful structural differences in the lactation yield performance of the different breeds that were studied. According to the findings that were obtained from the panel data analysis, several substantive conclusions can be drawn regarding the factors that influence milk yield in the goat breeds that were studied. It was determined that the age variable had a statistically significant effect on milk yield across the breeds. This finding indicates that milk yield increases as a function of increasing age of goats, at least within the range of ages that were observed in this study. However, this positive relationship between age and milk yield is valid only up to a certain age threshold, and it should be kept in mind that there may be a decrease in productive yield at later ages as animals move beyond their prime productive years. The relationship between age and productivity in livestock follows a curvilinear pattern, with production increasing during early maturity, reaching a peak during prime productive years, and then declining in advanced age. Similarly, it was observed that the extension of the lactation period significantly increases milk yield in a positive direction. This result reveals that longer lactation periods contribute to greater total milk production over the course of a lactation cycle. This makes biological sense because a longer lactation period provides more days during which milk is being produced, and thus greater cumulative milk yield. However, it should be noted that excessively long lactation periods may be associated with other management challenges and may not be optimal from an overall herd management perspective when considering factors such as rebreeding schedules and optimal calving intervals. A general increasing trend in milk yield was detected across the time period from 1991 to 1997. This temporal increase in milk yield may be the result of various factors including improvements in feeding conditions, improvements in management practices, genetic selection efforts that were ongoing during this time period, or improvements in animal health management. The upward trend suggests that there was progress being made in the productivity of these goat herds during the 7-year observation period. Such improvements in productivity over time are typical in livestock production systems where active management and improvement efforts are being implemented. In the comparisons that were made between the different breeds, it was observed that the Cukurova breed exhibited the highest fixed effect coefficient in terms of annual milk yield. This finding indicates that the Cukurova breed may possess a genetically superior milk production capacity compared to the other breeds that were included in this study. The fixed effects in a panel data model capture time invariant differences between the cross-sectional units, in this case the different breeds. A high fixed effect for the Cukurova breed indicates that this breed consistently produces more milk than the other breeds, holding constant the effects of age, lactation period, and time trends. This superior performance may be due to selective breeding efforts that have been focused on milk production traits in the Cukurova breed, or it may reflect the adaptation of this breed to the environmental and management conditions that prevailed in the study region. On the other hand, the milk yield of the pure Damascus breed was observed to be lower than the other breeds that were studied, and this breed had the lowest fixed effect coefficient. This suggests that the pure Damascus breed has a lower inherent milk production capacity compared to the other breeds, at least under the conditions that prevailed during this study. It is possible that the pure Damascus breed has been selected for other traits besides milk production, such as meat production or adaptation to particular environmental stresses, and consequently milk production may not be as high as in breeds that have been more intensively selected for dairy characteristics. It is also possible that the pure Damascus breed was not as well adapted to the specific environmental and management conditions that existed in the study location. The coefficient sizes of variables such as age and lactation length, which are factors that influence milk yield, provide quantitative information about the magnitude of the impact of these factors on production. The magnitude of the coefficients in the regression model indicates how much the dependent variable, milk yield, is expected to change for a unit change in each independent variable, holding all other variables constant. The elasticity coefficient for lactation length was found to be higher in magnitude compared to the elasticity coefficient for age. This finding suggests that the physiological dynamics of lactation are more decisive and have a larger impact on milk production than does the age of the animal. In other words, the length of the lactation period has a stronger effect on total milk yield than does the age of the animal. Conversely, the more limited but steady increase in the effect of age suggests that goats reach their highest milk yield capacity at a specific stage of maturity, and that yield may decline at later ages due to physiological regression in mammary tissue and other age-related changes in productive capacity. The relationship between age and milk yield is not linear throughout the entire lifespan of the animal. Young animals have not yet reached their full productive potential. Animals in their prime productive years have the highest yields, and older animals experience declining yields as physiological function deteriorates with advancing age. The significant differences in fixed effects between breeds highlight the critical role of genetic capacity in determining milk yield potential. The findings particularly demonstrate that the Cukurova goat has a higher milk production potential compared to other breeds that were included in this investigation. This superior genetic capacity for milk production makes the Cukurova breed an attractive option for dairy goat operations that are focused primarily on milk production as their main output. Producers who are selecting breeds for their operations should take into account these breed differences in milk yield potential when making decisions about which breeds to maintain in their herds. The choice of breed can have a substantial impact on the overall productivity and profitability of a dairy goat operation. The findings of this study that has been described in this lecture are consistent with similar studies that have been conducted and reported in the scientific literature. For example, in a study that was conducted that examined milk yield and milk composition of Saanen goats and German fawn goat crossbreeds, it was stated that both of these breeds showed high milk yield performance. The fact that the German fawn breed has been found to have high milk yield in multiple independent studies provides corroborating evidence for the reliability of this finding. In addition, in another study that was conducted, it was stated that the Cukurova breed was prominent in terms of milk yield compared to other breeds that were included in that comparison. This finding from the independent study provides external validation for the finding in the present study that the Cukurova breed has superior milk production capacity. Furthermore, in yet another study that was conducted, the behaviors of dairy goat breeds in hot climate conditions and cold climate conditions were examined specifically under the environmental conditions found in the Cukurova region, and it was stated that these breeds exhibited high adaptation abilities to varying environmental conditions. This finding regarding adaptation abilities can be evaluated as one potential explanation for why the Cukurova breed demonstrates advantages in terms of milk yield. Breeds that are well adapted to the local environmental conditions are likely to experience less physiological stress and are therefore able to allocate more resources to productive functions such as milk synthesis. The superior adaptation of the Cukurova breed to local conditions may thus contribute to its superior milk production performance. Another study compared the milk yield and milk components of Damascus goats and German fawn crossbred goats under Mediterranean climate conditions. In this study, both genotypes were reported to have high milk yields and similar milk composition characteristics. This result supports the finding of differences in milk yield performance between the breeds in the study that is being discussed in this lecture. The convergence of findings across multiple independent studies conducted in different locations and at different times strengthens confidence in the conclusions regarding breed differences in milk production capacity. As a result of these various findings that have been presented, important guidance and clues are provided for researchers and breeders who want to increase milk production in their goat herds. Choosing the right breed is very important in terms of milk yield potential, as the choice of breed will substantially influence production efficiency. Breed selection should be based on careful consideration of the production goals of the operation, the environmental conditions in which the animals will be raised, and the market demands for the products that will be produced. In addition to breed selection, careful planning of the lactation period and keeping the animals within the optimal production age range will further increase productive yield. It has been revealed through this research that managerial practices are equally important as determinants of milk production capacity as are genetic factors. Even breeds with superior genetic potential for milk production will not achieve their full productive capacity if they are not provided with appropriate nutrition, housing, healthcare, and other management inputs. Conversely, breeds with more modest genetic potential can achieve respectable levels of production if they are provided with excellent management. The interaction between genetic potential and environmental management conditions is an important consideration in livestock production systems. The panel data analysis methodology that was employed in this study proved to be an effective approach for analyzing livestock production data, where both temporal variation and cross-sectional variation are important features of the data structure. The ability of panel data methods to control for unobserved heterogeneity between cross-sectional units while simultaneously modeling time trends makes these methods particularly well suited for livestock production research. The fixed effects model that was selected based on the Hausman test allowed the estimation of breed-specific intercepts that capture the time-invariant differences in milk production capacity between the breeds. The statistical testing procedures that were employed to verify that the assumptions of the regression model were satisfied, including tests for multicollinearity, heterosedasticity, and autocorrelation, and the use of corrected standard errors to address violations of assumptions, all contribute to confidence in the validity and reliability of the parameter estimates that were obtained. When regression assumptions are violated, parameter estimates may remain unbiased, but standard errors may be incorrect, leading to invalid inference. The use of panel-corrected standard errors addresses this problem and provides valid statistical inference even in the presence of heteroscedasticity and autocorrelation. The careful attention to preliminary diagnostic testing, including tests for cross-sectional dependence and slope heterogeneity, ensures that the appropriate model specification and estimation procedures were employed. Different configurations of cross-sectional dependence and slope heterogeneity require different unit root testing procedures and different estimation approaches. By conducting comprehensive diagnostic testing before proceeding to model estimation, this study avoided potential specification errors that could have led to biased or inconsistent parameter estimates. The finding that lactation length has a larger effect on milk yield than does age has practical implications for herd management. Producers can potentially influence lactation length through breeding and management decisions, whereas age structure of the herd changes more slowly and is constrained by biological and economic factors. Strategies to extend lactation length in high-producing animals may be an effective approach to increasing overall milk production from the herd. However, such strategies must be balanced against other management considerations such as maintaining appropriate calving intervals and avoiding excessive metabolic stress on high-producing animals. The positive effect of age on milk yield, up to a certain point, reflects the biological reality that young animals are still growing and developing their productive capacity, while mature animals have fully developed mammary systems and have reached their peak productive potential. However, the eventual decline in productivity with advanced age means that maintaining an optimal age structure in the herd is important for maximizing overall herd productivity. Culling decisions should take into account both the current productive status of individual animals and their expected future productive trajectory based on their age. The temporal trend toward increasing milk yield over the 7-year study period suggests that continuous improvement in production is achievable through ongoing selection and management efforts. This finding should be encouraging to producers and researchers who are working to improve productivity in goat production systems. Even modest rates of genetic gain, when sustained over multiple years, can result in substantial cumulative improvements in productive capacity. However, it is important to recognize that genetic improvement must be accompanied by corresponding improvements in nutrition, health management, and other inputs in order to allow animals to express their improved genetic potential. The breed differences that were identified in this study highlight the importance of matching breed selection to production objectives and environmental conditions. Breeds that perform well under one set of conditions may not perform as well under different conditions. The Cukurova breed appears to be particularly well suited to milk production under the conditions that prevailed in this study, but this does not necessarily mean that this breed would be the optimal choice under all possible circumstances. Breed selection decisions should be based on careful evaluation of the specific circumstances of each production operation. The lower performance of the pure Damascus breed in this study does not necessarily indicate that this is an inferior breed in all respects. Different breeds have been selected for different traits and may excel in different production systems or under different market conditions. A breed that is not optimal for maximum milk production may have advantages in terms of other traits such as meat production, fiber production, adaptability to harsh environments, disease resistance, or other characteristics that may be valued in different production contexts. A comprehensive evaluation of breed performance should consider multiple traits and not focus exclusively on a single production characteristic. The methodology of panel data analysis that has been demonstrated in this study can be applied to other questions and other datasets in animal production research. Whenever data are available that have both a cross-sectional dimension and a time dimension, panel data methods should be considered as a potentially superior alternative to conventional cross-sectional or time series methods. The ability to control for unobserved heterogeneity and to estimate more efficient parameters by pooling information across both dimensions of the data makes panel methods particularly attractive for agricultural and animal science applications where sample sizes are often limited and data collection is expensive. The adjusted R-squared values that were obtained in this study, ranging from approximately 0.4 to 0.64, indicate that the models explain a substantial but not complete proportion of the variation in milk yield. This is typical for biological systems where many factors influence outcomes and considerable random variation exists even after accounting for measured explanatory variables. The unexplained variation may be due to unmeasured environmental factors, individual animal variation in genetic potential beyond breed effects, measurement error, and inherent stochasticity in biological processes. Future research might seek to identify additional measurable factors that could be included in models to explain more of the variation in milk yield. One limitation of this study is that it was conducted during a specific time period, from 1991 to 1997, and conditions may have changed since that time. Genetic selection has continued in these breeds over the subsequent decades, and management practices have likely evolved. The specific magnitudes of breed differences and the effects of age and lactation length that were estimated in this study may not be exactly the same if similar data were collected and analyzed today. However, the general patterns and relationships that were identified are likely to remain relevant and applicable. Another consideration is that this study was conducted in a specific geographic region, Turkey, and the results may not be directly generalizable to other regions with different environmental conditions, different management systems, or different market structures. Breed performance is often context-dependent, and a breed that performs well in one environment may not perform as well in a different environment. Caution should be exercised in extrapolating these findings to substantially different production contexts without appropriate validation. Despite these limitations, this study makes important contributions to the scientific literature on goat milk production and demonstrates the value of panel data analysis methods in animal production research. The specific findings regarding breed differences, the effects of age and lactation length, and the temporal trends in productivity provide useful information for producers and policymakers. The methodological demonstration of the application of panel data methods, including comprehensive diagnostic testing and appropriate model selection procedures, provides a template that can be followed by other researchers working with similar types of data. Future research might extend this analysis in several directions. One possibility would be to collect more recent data and examine whether the breed differences and the effects of explanatory variables have changed over time as genetics and management have evolved. Another possibility would be to expand the set of explanatory variables to include measures of nutrition, health status, environmental conditions, and management practices, to obtain a more complete understanding of the factors that influence milk yield. Yet another possibility would be to examine other outcome variables in addition to milk yield, such as milk composition, reproduction performance, or economic returns, to obtain a more comprehensive evaluation of breed performance. The data collection effort that underlies this study involves systematic recording of milk yield, lactation period, and age information for randomly selected individuals from each breed over a 7-year period. Such long-term systematic data collection requires substantial commitment of resources and careful attention to data quality. The availability of such high-quality longitudinal data is what makes sophisticated statistical analyses like the panel data methods employed in this study possible. Continued investment in livestock data collection systems is important for supporting research that can contribute to improved productivity and sustainability of animal production systems. The random selection of individual animals for data collection helps to ensure that the sample is representative of the larger population of animals in each breed, which supports the generalizability of the findings. If animals had been selected based on their production levels or other characteristics, the sample could have been biased and the results might not be representative of typical performance in these breeds. Random sampling is an important principle of sound research design that supports valid statistical inference. The use of standardized measurement protocols for milk yield, lactation period, and age is important for ensuring data quality and comparability across animals and across time. Consistent measurement procedures reduce measurement error and increase the reliability of the data. Quality control procedures to verify data accuracy and identify potential errors or outliers are important components of sound data management in research studies. The credibility of research findings depends fundamentally on the quality of the underlying data. The statistical software package of use version 9 that was used for the panel data analysis is a widely used and well-validated tool for econometric analysis. The availability of reliable statistical software has made sophisticated analytical methods accessible to researchers who might not have the expertise to program these methods from scratch. However, users of statistical software must understand the underlying statistical theory and assumptions in order to properly interpret results and avoid misapplication of methods. Statistical software is a tool that must be used appropriately and thoughtfully. The theoretical framework that underlies panel data analysis recognizes that observations are not independent random samples, but rather have structure arising from the panel design where multiple observations are collected on each cross-sectional unit. This within-unit correlation must be properly accounted for in the statistical analysis. The fixed effects model accomplishes this by allowing each cross-sectional unit to have its own intercept, which captures time-invariant characteristics of that unit. By removing these unit-specific effects, the fixed effects model estimates the effects of the time-varying explanatory variables on the outcome variable. The random effects model is an alternative approach that treats the unit-specific effects as random variables rather than as fixed parameters. This approach is more efficient when the assumptions are satisfied, but it requires the assumption that the unit-specific effects are uncorrelated with the explanatory variables. The Hausman test examines whether this assumption is satisfied. When the Hausman test rejects the null hypothesis, as it did in this study, this indicates that the random effects assumption is not satisfied and the fixed effects model should be preferred. The fixed effects model produces consistent estimates even when the unit-specific effects are correlated with the explanatory variables, though at some cost in terms of efficiency. The attention to diagnostic testing for heterosedasticity and autocorrelation, and the use of panel-corrected standard errors to address these issues, reflects current best practices in applied econometric analysis. Early applications of panel data methods sometimes neglected these issues, which could lead to invalid inference even when parameter estimates were consistent. The recognition that heterosedasticity and autocorrelation are common in panel data and the development of methods to address these issues represent important methodological advances that have improved the reliability of applied econometric research. Cross-sectional dependence is another issue that has received increasing attention in the panel data literature in recent years. When cross-sectional units are subject to common shocks or when there are spillover effects between units, the assumption of cross-sectional independence is violated. This can affect both the properties of unit root tests and the properties of parameter estimators. Second-generation unit root tests and other methods that allow for cross-sectional dependence have been developed to address these issues. The application of tests for cross-sectional dependence in this study and the use of appropriate methods conditional on the results of these tests reflects awareness of these methodological issues. The issue of slope heterogeneity addresses whether the effects of explanatory variables on the outcome variable are the same across all cross-sectional units or whether they differ. If slopes are heterogeneous but are treated as homogeneous in the analysis, parameter estimates may be biased and inference may be invalid. Testing for slope heterogeneity and using appropriate methods conditional on the results is another important aspect of sound panel data analysis. This study tested for slope heterogeneity and found that some variables exhibited homogeneous slopes while others exhibited heterogeneous slopes, and appropriate methods were employed in each case. The econometric theory underlying fixed effects estimation in panel data models is based on the within transformation, which differences out the unit-specific effects by subtracting the unit-specific mean from each observation. This transformation removes all time-invariant characteristics of each unit, whether observed or unobserved. After applying the within transformation, ordinary least squares estimation of the transformed model yields consistent estimates of the slope parameters. The loss of information about time-invariant characteristics means that fixed effects models cannot estimate the effects of variables that do not change over time within units. But this is an acceptable trade-off to obtain consistent estimates of the effects of time-varying variables without requiring strong assumptions about the correlation between unit effects and explanatory variables. The standard errors that are computed in fixed effects estimation must account for the panel structure of the data. If standard errors are computed as if observations were independent, they will be incorrect because observations on the same unit are correlated through the unit-specific effect. Clustered standard errors that allow for arbitrary correlation within units but assume independence across units are commonly used to address this issue. Panel-corrected standard errors go further by also allowing for heteroscedasticity across units and contemporaneous correlation across units. These more sophisticated standard error estimators provide more accurate inference in the presence of complex error structures that are common in panel data. The interpretation of regression coefficients in logarithmic specifications differs from the interpretation in linear specifications. When both a dependent variable and an independent variable are in logarithms, the coefficient represents an elasticity, which is the percentage change in the dependent variable associated with a 1% change in the independent variable. When only the dependent variable is in logarithms, the coefficient must be transformed to obtain the percentage effect. When only an independent variable is in logarithms, the coefficient represents the change in the dependent variable associated with a 1% change in the independent variable. Careful attention to the functional form specification is important for correct interpretation of results. The assumption of strict exogeneity in fixed effects models requires that the error term at time t is uncorrelated with the explanatory variables at all time periods, not just at time t. This is a stronger assumption than the contemporaneous exogeneity assumption that is required for cross-sectional regression. Violations of strict exogeneity can occur when there are lagged dependent variables, when there is feedback from past outcomes to current explanatory variables, or when there is measurement error in explanatory variables. When strict exogeneity is violated, fixed effects estimates are inconsistent, and alternative estimators such as instrumental variables methods may be required. The number of observations in a panel dataset is the product of the number of cross-sectional units and the number of time periods. In this study, with 5 breeds and 7 years, there are 35 total observations assuming complete data for all breed year combinations. However, the effective sample size for estimating the effects of time-varying variables is determined primarily by the within-unit variation over time, not by the total number of observations. With only 7 time periods per unit, the precision of estimates is limited by the amount of time series variation in each breed. Longer panels with more time periods per unit generally allow more precise estimation of dynamic relationships. The balanced panel structure, where all units are observed in all time periods, is advantageous for several reasons. Balanced panels simplify estimation and inference because there are no complications from varying numbers of observations per unit. Balanced panels also avoid potential selection bias that can arise if missingness is related to the outcome variable or to unobserved characteristics. However, requiring a balanced panel can result in discarding useful information if some units are not observed in all periods. The choice between maintaining balance and including all available information involves trade-offs that depend on the specific circumstances of the research problem. The assumption that the number of cross-sectional units is large is important for the asymptotic properties of panel estimators. Fixed effects estimators are consistent as the number of cross-sectional units approaches infinity, holding the number of time periods fixed. In contrast, the properties of fixed effects estimators as the number of time periods approaches infinity, holding the number of cross-sectional units fixed, are different and depend on additional assumptions about the time series properties of the data. In this study with only 5 cross-sectional units, asymptotic theory based on large cross-sectional dimension may not provide accurate approximations to the finite sample properties of the estimators. This is a limitation that should be recognized when interpreting the results. The treatment of time effects in panel data models is an important specification choice. If there are shocks or trends that affect all units similarly in each time period, these common time effects should be controlled for to avoid omitted variable bias. Time effects can be controlled by including time dummy variables, which allow the intercept to vary across time periods, or by including time trends or other time-varying variables that capture common temporal patterns. The specification of time effects should be based on theoretical considerations about the nature of temporal variation in the outcome variable and on empirical testing of alternative specifications. The distinction between pooled ordinary least squares estimation, random effects estimation, and fixed effects estimation is fundamental in panel data analysis. Pooled ordinary least squares treats all observations as independent and does not account for the panel structure. This approach is appropriate only if there are no unit-specific effects or if the unit-specific effects are uncorrelated with the explanatory variables and the error structure satisfies the classical assumptions. Random effects estimation models the unit-specific effects as random variables and uses generalized least squares to exploit the structure of the error covariance matrix. Fixed effects estimation removes the unit-specific effects through transformation and uses ordinary least squares on the transformed data. The efficiency of random effects estimation relative to fixed effects estimation depends on whether the random effects assumptions are satisfied. If the unit-specific effects are uncorrelated with the explanatory variables, random effects is more efficient because it uses both between unit variation and within unit variation, whereas fixed effects uses only within unit variation. However, if the unit-specific effects are correlated with the explanatory variables, random effects is inconsistent whereas fixed effects remains consistent. The Hausman test exploits this difference to test whether random effects or fixed effects should be preferred. A statistically significant Hausman test statistic indicates that the random effects assumptions are rejected and fixed effects should be used. The interpretation of fixed effects in panel data models depends on the context of the analysis. In this study, the fixed effects represent breed-specific intercepts that capture all time-invariant differences between breeds. These differences could include genetic characteristics, historical factors, average environmental conditions experienced by each breed, or any other characteristics that distinguish the breeds but do not vary over time. The fixed effects capture these influences even though they are not explicitly measured or included as variables in the model. This is one of the major advantages of fixed effects models over cross-sectional models where unmeasured time-invariant characteristics would create omitted variable bias. The magnitude of the fixed effects estimates provides information about the relative positions of the different breeds. Large positive fixed effects indicate breeds with systematically higher milk yield, while large negative fixed effects indicate breeds with systematically lower milk yield, after controlling for the effects of age, lactation period, and time trends. The differences in fixed effects between breeds reflect fundamental differences in milk production capacity that are attributable to breed characteristics. These differences are economically meaningful and provide guidance for breed selection decisions in dairy goat operations. The coefficient on age in the milk yield regression reflects the average relationship between age and milk yield across all breeds, assuming that this relationship is homogeneous across breeds. If the relationship between age and milk yield differs across breeds, this could be investigated by interacting the age variable with breed dummy variables. Such interactions would allow the slope of the age effect to differ by breed. However, with limited sample size, estimating breed-specific age effects may not be feasible due to multicollinearity and insufficient degrees of freedom. The assumption of homogeneous age effects is a simplification that may or may not be appropriate depending on the true underlying relationships. Similarly, the coefficient on lactation period reflects the average relationship between lactation period and milk yield across all breeds. The positive coefficient indicates that longer lactation periods are associated with higher cumulative milk yield, which is expected because more days of lactation provide more opportunity for milk production. The magnitude of this coefficient quantifies this relationship and can be used to predict how much additional milk yield would be expected from extending the lactation period by a given number of days. Such predictions can inform management decisions about optimal lactation length from a production standpoint, though other factors such as reproduction and animal health must also be considered. The time trend in milk yield that was detected in this study could arise from several sources. Genetic improvement through selective breeding would tend to increase milk yield over time as superior animals are selected for breeding and inferior animals are culled. Improvements in nutrition, housing, healthcare, and other management practices would also tend to increase milk yield over time. Changes in the composition of the herds, such as shifts in the age distribution toward prime productive ages, could also contribute to temporal trends. Distinguishing between these different sources of temporal change would require additional data on breeding decisions, management practices, and herd composition. The functional form of the relationship between age and milk yield is an important consideration that affects the interpretation of the estimated age coefficient. If the relationship is linear, the coefficient represents the constant change in milk yield per year of age. However, biological considerations suggest that the relationship is likely to be non-linear, with milk yield increasing during early maturity, reaching a peak at some optimal age, and then declining in old age. A quadratic specification, which includes both age and age squared, can capture such a curvilinear relationship. Testing alternative functional forms and selecting the specification that best fits the data is an important part of model specification. The residuals from the regression represent the portion of milk yield that is not explained by the included explanatory variables. Analysis of residuals can provide diagnostic information about model specification and about violations of regression assumptions. Plots of residuals against fitted values can reveal heterosedasticity or nonlinearity. Plots of residuals against individual explanatory variables can reveal misspecification of functional form. Plots of residuals over time can reveal autocorrelation or structural breaks. Normal probability plots of residuals can reveal departures from normality of the error distribution. Comprehensive residual analysis is an important step in validating the adequacy of the fitted model. The statistical significance of individual coefficients is assessed using t-tests based on the ratio of the coefficient estimate to its standard error. Under the null hypothesis that the true coefficient is 0, this ratio follows a t-distribution with degrees of freedom equal to the number of observations minus the number of parameters being estimated. If the absolute value of the t-statistic exceeds the critical value from the t-distribution at the chosen significance level, the null hypothesis is rejected and the coefficient is deemed statistically significant. Statistical significance indicates that the observed relationship is unlikely to have arisen by chance if the true relationship was 0, but does not necessarily indicate practical or economic significance. The overall significance of the regression model is assessed using an f-test that tests the null hypothesis that all slope coefficients are simultaneously equal to 0. If this null hypothesis is rejected, it indicates that at least one of the explanatory variables has a statistically significant relationship with the dependent variable. The f-statistic is constructed from the ratio of explained variation to unexplained variation, adjusted for degrees of freedom. A large f-statistic and small p-value indicate strong evidence against the null hypothesis of no relationship. The f-test provides an overall assessment of model significance that complements the individual t-tests for specific coefficients. The adjusted R-squared statistic measures the proportion of variation in the dependent variable that is explained by the model, adjusted for the number of parameters being estimated. Unlike the unadjusted R-squared, which necessarily increases when additional variables are added to the model regardless of their true explanatory power, the adjusted R-squared can decrease if added variables do not sufficiently improve model fit to justify the loss of degrees of freedom. The adjusted R-squared provides a measure of overall model fit that penalizes model complexity and is therefore useful for comparing models with different numbers of explanatory variables. The Durbin-Watson statistic is a test statistic for detecting the presence of autocorrelation in the residuals from a regression analysis. Autocorrelation violates the assumption of independent errors and can lead to inefficient estimates and invalid standard errors. The Durbin-Watson statistic ranges from 0 to 4, with a value near 2 indicating no autocorrelation, values near 0 indicating positive autocorrelation, and values near 4 indicating negative autocorrelation. Formal hypothesis tests can be conducted using critical values from the Durbin-Watson distribution. Detection of autocorrelation indicates the need for corrective measures such as using autocorrelation consistent standard errors or modeling the autocorrelation structure explicitly. The Broich-Pagan test is a test for heterosedasticity. which is the condition where the variance of the error term differs across observations. Heterosedasticity violates the assumption of constant error variance and leads to inefficient estimates and incorrect standard errors in ordinary least squares regression. The Breusch-Pagan test regresses the squared residuals on the explanatory variables and tests whether the coefficients are jointly significant. A significant test statistic indicates the presence of heterosedasticity. When heterosedasticity is detected, robust standard errors or weighted least squares methods should be used to obtain valid inference. The assumption of normally distributed errors is not required for consistency or unbiasedness of ordinary least squares estimates, but it is required for exact finite sample validity of standard hypothesis tests based on the T and F distributions. In large samples, the central limit theorem provides justification for approximate normality of test statistics even when errors are not normally distributed. However, in small samples, departures from normality can affect the validity of inference. Tests for normality, such as the Jark-Berra test, can be used to assess whether the normality assumption is reasonable. Substantial departures from normality may indicate model misspecification or the presence of outliers. Outliers and influential observations can have substantial effects on regression results, particularly in small samples. An outlier is an observation with an unusually large residual, indicating that it is poorly fit by the model. An influential observation is one whose inclusion or exclusion substantially changes the regression results. Various diagnostic statistics such as studentized residuals, leverage statistics, Cook's distance, and DF betas can be used to identify outliers and influential observations. Investigation of such observations can reveal data errors, unusual circumstances, or model misspecification. Sensitivity analysis that examines how results change when influential observations are excluded can assess the robustness of conclusions. The interpretation of causality in observational studies requires careful consideration of potential confounding factors and the direction of causation. Regression analysis identifies associations between variables but does not by itself establish causal relationships. To interpret estimated coefficients as causal effects requires the assumption that all confounding variables are controlled for in the model and that the direction of causation runs from the explanatory variables to the dependent variable rather than the reverse. In experimental settings with random assignment, causal interpretation is straightforward. In observational settings, establishing causality requires theoretical arguments, careful research design, and sensitivity to potential threats to causal inference. The issue of reverse causation is a potential concern in studies of animal production where current outcomes may influence future inputs. For example, animals with high milk yield may be given preferential treatment in terms of feeding or other management practices, creating reverse causation from milk yield to management inputs. If such feedback effects are present and not properly controlled for, estimated coefficients may be biased. Instrumental variables methods or dynamic panel data methods that explicitly model the feedback process may be needed to obtain consistent estimates in the presence of reverse causation. The generalizability of research findings to populations and settings beyond those studied is an important consideration in evaluating the external validity of research. Results from a study conducted in one region during one time period with particular breeds may not apply to other regions, time periods, or breeds. Differences in environmental conditions, management systems, genetic background, or market structures could lead to different relationships between variables. Replication of findings across multiple studies in diverse settings strengthens confidence in generalizability. Meta-analysis methods can be used to synthesize findings across multiple studies and assess the consistency of relationships. The economic interpretation of statistical results requires translating coefficient estimates and statistical significance into practical implications for decision-making. A statistically significant effect may be economically insignificant if the magnitude is too small to justify changes in practice or if the costs of implementing changes exceed the benefits. Conversely, An effect that is not statistically significant may still be economically important if the point estimate suggests substantial impacts, even though precision is limited by sample size. Cost-benefit analysis that weighs the expected gains from implementing research findings against the costs of implementation is essential for translating research into practice. The role of uncertainty in decision-making must be recognized when applying research findings to practical situations. Parameter estimates are subject to sampling variability, and confidence intervals provide a range of plausible values for the true parameters. When the confidence interval is wide, there is substantial uncertainty about the true effect, and decisions based on point estimates alone may be risky. Decision-making under uncertainty may require considering worst-case scenarios, sensitivity analysis, or formal decision-theoretic approaches that explicitly incorporate uncertainty. Risk aversion may lead decision-makers to prefer conservative strategies when facing substantial uncertainty about outcomes. The interaction between research and practice is a two-way process where research findings inform practice and practical experience raises questions for research. Applied research that addresses questions arising from practical problems is more likely to generate findings that are useful for improving practice. Engagement between researchers and practitioners throughout the research process, from problem identification through study design to interpretation and dissemination of results, enhances the relevance and impact of research. Extension services and other mechanisms for translating research into practice play important roles in ensuring that research findings reach and influence practitioners. The long-term sustainability of livestock production systems depends on balancing productivity goals with environmental, social, and economic considerations. Focusing exclusively on maximizing production without regard to environmental impacts, animal welfare, or economic viability is not sustainable in the long run. Research that considers multiple dimensions of sustainability and examines trade-offs between competing objectives can contribute to the development of production systems that meet current needs without compromising the ability of future generations to meet their needs. Integrated assessment frameworks that combine biological, environmental, economic, and social dimensions provide comprehensive evaluation of livestock production systems. The role of policy and institutions in shaping livestock production systems should not be overlooked. Government policies related to agricultural subsidies, environmental regulations, animal health programs, trade policies, and rural development can all influence the structure and performance of livestock production. Research on the impacts of policies and on the design of more effective policies can inform policy debates and improve policy outcomes. Institutional factors such as property rights, market structure, access to credit, and availability of technical assistance also affect production decisions and outcomes. Understanding these institutional factors is important for designing interventions that effectively address constraints to productivity and sustainability. The adoption of new technologies and practices by livestock producers depends on various factors, including the characteristics of the innovation, the characteristics of potential adopters, and the social and institutional context. Innovations that offer clear advantages in terms of productivity or profitability, that are compatible with existing practices and values, that are simple to understand and implement, and that can be tried on a limited scale before full adoption are more likely to be adopted. Understanding the factors that influence adoption can help in designing innovations and extension programs that are more likely to achieve widespread uptake and impact. The heterogeneity among livestock producers in terms of resources, goals, constraints, and contexts means that one-size-fits-all recommendations are unlikely to be appropriate for all producers. Some producers may prioritize maximizing production, while others may prioritize minimizing costs, managing risk, ensuring sustainability, or achieving multiple objectives simultaneously. Research that acknowledges this heterogeneity and provides decision support tools that can be adapted to diverse circumstances is more useful than research that prescribes uniform practices. Participatory research approaches that involve producers in identifying problems, designing studies, and interpreting results can enhance the relevance and uptake of research findings. The advancement of statistical and computational methods continues to expand the range of questions that can be addressed and the sophistication of analyses that can be performed. The availability of more powerful computers and more advanced software has made it feasible to estimate complex models that would have been computationally prohibitive in earlier eras. New econometric methods continue to be developed to address challenging problems such as endogeneity, measurement error, missing data, and complex error structures. Staying current with methodological developments and applying appropriate methods to research questions enhances the quality and credibility of research. The importance of data quality cannot be overstated in empirical research. No amount of sophisticated statistical analysis can compensate for poor quality data. Investment in careful data collection, quality control procedures, documentation of data sources and methods, and transparent reporting of data limitations is essential for credible research. Sharing data with other researchers enhances transparency, enables replication and validation of findings, and allows secondary analyses that can extract additional insights from existing data. Data management practices that ensure long-term preservation and accessibility of research data are important for building cumulative knowledge. The theoretical foundations of econometric methods rest upon probability theory and statistical inference. The regression model is fundamentally a probabilistic model that describes the conditional distribution of the dependent variable given the explanatory variables. The error term represents all factors that affect the dependent variable but are not included as explanatory variables. The assumptions about the error term, including its mean, variance, correlation structure, and distribution, determine the properties of estimators and the validity of inference procedures. Understanding these theoretical foundations is essential for proper application and interpretation of econometric methods. The concept of statistical power refers to the probability of detecting a true effect when it exists. Low statistical power, which can result from small sample sizes or high variability, means that true effects may fail to be detected as statistically significant. The consequences of low power include an increased risk of type 2 errors, where false null hypotheses are not rejected, and reduced precision of estimates as reflected in wide confidence intervals. Power analysis can be conducted prior to data collection to determine the sample size needed to achieve adequate power for detecting effects of a specified magnitude. Post hoc power analysis, while sometimes controversial, can provide insight into the limitations of studies that fail to detect significant effects. The distinction between statistical significance and practical significance is important but sometimes overlooked. Statistical significance indicates that an observed relationship is unlikely to have arisen by chance, but it does not necessarily indicate that the relationship is large enough to be important in practice. In large samples, even trivial effects can be statistically significant. Conversely, in small samples, important effects may fail to achieve statistical significance due to limited power. Interpretation of results should consider both statistical significance and practical significance, with emphasis on effect sizes and confidence intervals in addition to p-values. The problem of multiple testing arises when many hypothesis tests are conducted simultaneously or sequentially. When multiple tests are performed, the probability that at least one test will yield a false positive result increases with the number of tests. Various corrections for multiple testing, such as the Bonferroni correction or the false discovery rate approach, can be applied to control the overall error rate. However, these corrections reduce power and may be overly conservative in some contexts. The appropriate approach to multiple testing depends on the research context and the relative costs of type 1 and type 2 errors. The issue of publication bias arises when studies with statistically significant results are more likely to be published than studies with null or inconclusive results. This selective publication can create a distorted picture of the evidence base if published studies are not representative of all studies that have been conducted. Meta-analyses that synthesize published studies may be biased if publication bias is present. Methods for detecting and correcting for publication bias, such as funnel plots and trim and fill procedures, can be applied, but these methods have limitations. Pre-registration of studies and publication of null results can help address publication bias. The debate over frequentist versus Bayesian approaches to statistical inference reflects fundamentally different conceptualizations of probability and different approaches to incorporating prior information. Frequentist methods treat parameters as fixed unknown constants and interpret probability in terms of long-run frequencies. Bayesian methods treat parameters as random variables with probability distributions and update prior beliefs based on data using Bayes' theorem. Each approach has advantages and disadvantages, and the choice between them depends on philosophical preferences, practical considerations, and the specific characteristics of the research problem. The role of assumptions in statistical analysis requires careful consideration. All statistical methods rest on assumptions about the data-generating process, and violations of these assumptions can affect the validity of inferences. However, the degree to which violations matter depends on the specific assumption, the magnitude of the violation, and the sample size. Some procedures are robust to violations of certain assumptions, meaning that they perform adequately even when assumptions are not strictly satisfied. Understanding which assumptions are critical and which are less important helps in assessing the reliability of results and in choosing appropriate methods. The concept of identification in econometrics refers to whether the parameters of interest can be uniquely determined from the observable data. A model is identified if different parameter values lead to different observable distributions. Identification problems arise when the structure of the model or the available data does not provide enough information to distinguish between alternative parameter values. Addressing identification requires theoretical restrictions, additional data, or alternative estimation strategies. Understanding identification issues is fundamental to credible econometric analysis. The interpretation of interaction terms in regression models requires careful attention to avoid common errors. An interaction between two variables means that the effect of one variable on the outcome depends on the value of the other variable. The coefficient on an interaction term represents the change in the slope of one variable as the other variable increases by 1 unit. When interactions are present, the marginal effect of a variable is not simply its coefficient but must be calculated taking into account the interaction term. Graphical displays of predicted values at different combinations of interacting variables often aid interpretation. The choice of functional form for regression models affects both the interpretation of coefficients and the fit of the model to the data. Linear models assume that the relationship between variables is additive and constant across the range of the data. Logarithmic transformations can be used to model multiplicative relationships or constant elasticities. Polynomial terms can capture nonlinear relationships while maintaining a linear in-parameters structure. More flexible functional forms such as splines or non-parametric methods can accommodate complex patterns but may be more difficult to interpret and may suffer from overfitting in small samples. The issue of overfitting arises when a model fits the particular sample too closely, capturing noise rather than systematic patterns. Overfit models perform well in sample but poorly out of sample because they do not generalize to new data. The risk of overfitting increases with model complexity relative to sample size. Methods for addressing overfitting include regularization techniques that penalize model complexity, cross-validation procedures that assess out-of-sample performance, and information criteria that balance fit and parsimony. Finding the appropriate balance between model flexibility and generalizability is a central challenge in statistical modeling. The concept of degrees of freedom in statistical analysis relates to the amount of independent information available for estimation. Each parameter that is estimated consumes 1 degree of freedom. The residual degrees of freedom, which equal the sample size minus the number of estimated parameters, determine the precision of estimates and the distribution of test statistics. Insufficient degrees of freedom can lead to imprecise estimates and low statistical power. In complex models with many parameters relative to sample size, parameter estimates may be unstable and results may be unreliable. The extrapolation of regression results beyond the range of the observed data is hazardous and should be done with caution. Relationships that hold within the observed range may not hold outside that range. For example, a linear relationship within the observed range does not imply that the relationship remains linear at extreme values. Predictions based on extrapolation carry greater uncertainty than predictions within the range of the data. When extrapolation is necessary, it should be acknowledged explicitly, and appropriate caution should be exercised in interpreting results. The aggregation of data from individual units to group-level averages can affect the relationships that are observed. Ecological fallacy refers to the error of inferring individual-level relationships from group-level data. Simpson's paradox refers to situations where a relationship observed in aggregated data reverses or disappears when data are disaggregated. These phenomena highlight the importance of analyzing data at the appropriate level of aggregation for the research question. Multilevel or hierarchical models can explicitly account for structure at multiple levels of aggregation. The temporal aggregation of data, such as using annual averages when underlying variation occurs at higher frequencies, can obscure relationships or create spurious patterns. Time aggregation can smooth out short-term fluctuations that may be of interest or may contain important information about underlying processes. The choice of temporal aggregation should be based on theoretical considerations about the timescale at which relationships operate and on practical considerations about data availability. High-frequency data allow finer resolution of temporal dynamics, but may require more complex methods to handle serial correlation and other complications. The concept of mean reversion refers to the tendency for extreme values to be followed by values closer to the mean. In the context of livestock production, animals with unusually high production in one period may show lower production in subsequent periods simply due to regression toward the mean rather than due to any systematic change in their productive capacity. Failure to account for mean reversion can lead to incorrect inferences about the effects of interventions or the persistence of performance differences. Understanding natural variability and mean reversion is important for proper interpretation of longitudinal data. The autocorrelation structure in time series data affects both the efficiency of estimators and the validity of standard errors. Positive autocorrelation, where successive observations tend to be similar, is common in economic and biological time series. Negative autocorrelation, where successive observations tend to alternate, is less common but can occur in some contexts. The appropriate treatment of autocorrelation depends on whether it is considered a nuisance that should be corrected for or a substantive phenomenon that should be modeled. Autoregressive or moving average models can explicitly represent autocorrelation structure. The specification of lag structure in dynamic models, where current outcomes depend on past values of variables, requires balancing theoretical considerations against empirical evidence and degrees of freedom constraints. Including too few lags may omit relevant information and lead to misspecification. Including too many lags may waste degrees of freedom and introduce multicollinearity. Information criteria or sequential testing procedures can guide the selection of appropriate lag length. The distributed lag pattern, showing how effects accumulate over time, provides insight into the dynamics of adjustment processes. The treatment of seasonality in time series data is important in agricultural and livestock applications where production follows seasonal patterns. Seasonal patterns can be modeled using seasonal dummy variables, seasonal moving averages, or other methods. Failure to account for seasonality can lead to spurious findings if seasonal patterns are confounded with other variables of interest. Seasonal adjustment procedures can remove seasonal components to focus on underlying trends and cycles. However, seasonal adjustment should be done carefully to avoid removing substantive variation of interest. The concept of co-integration in time series analysis refers to the existence of long-run equilibrium relationships between non-stationary variables. Even though individual series may trend over time, linear combinations of co-integrated series are stationary. Testing for co-integration and estimating co-integrating relationships using methods such as error correction models allows the analysis of both short-run dynamics and long-run equilibria. Co-integration methods are particularly relevant for analyzing relationships between economic variables that share common trends. The evaluation of forecasting models involves both in-sample and out-of-sample assessments. In-sample fit measures such as R-squared or information criteria assess how well the model explains observed data but may overstate performance due to overfitting. Out-of-sample forecast accuracy provides a more stringent test of model performance because it evaluates predictions for data not used in estimation. Common forecast accuracy measures include mean absolute error, root mean squared error, and mean absolute percentage error. Forecast encompassing tests can compare the performance of competing models. The uncertainty in forecasts increases with the forecast horizon because errors accumulate over time and because the underlying relationships may change. Forecast intervals that quantify uncertainty should account for both parameter uncertainty and fundamental uncertainty about future disturbances. Scenario analysis that examines forecasts under different assumptions about future conditions can provide insight into the sensitivity of forecasts to key drivers. Probabilistic forecasting that provides full probability distributions rather than point forecasts can support better decision-making under uncertainty. The communication of statistical results to non-technical audiences requires translating technical findings into accessible language while maintaining accuracy. Effective communication involves explaining what was studied and why, describing the methods in general terms without excessive jargon, presenting key findings clearly with appropriate visualization, acknowledging limitations and uncertainties, and drawing out practical implications. Graphical displays such as plots of predicted values or effect sizes with confidence intervals often communicate results more effectively than tables of regression coefficients. Clear communication enhances the impact and utilization of research. The ethical responsibilities of researchers include conducting research with integrity, reporting results honestly and accurately, acknowledging limitations and uncertainties, treating research subjects appropriately, managing conflicts of interest, and using resources responsibly. Ethical guidelines and institutional review boards provide frameworks for ensuring that research meets ethical standards. Transparency in reporting methods and results allows others to evaluate and replicate research. Responsible conduct of research builds trust in the scientific enterprise and ensures that research serves the public good. The cumulative nature of scientific knowledge means that individual studies contribute to an evolving body of evidence. Replication of findings across multiple studies and synthesis of evidence through meta-analysis and systematic review strengthen confidence in conclusions. Contradictory findings across studies may reflect differences in populations, methods, or contexts, and can stimulate further research to resolve discrepancies. The scientific process involves an ongoing dialogue between theory and evidence, with theories being refined and sometimes overturned as new evidence accumulates. The limitations of this study must be acknowledged in interpreting the findings and considering their applicability. The relatively small number of breeds and the limited time period constrain the generalizability of results. The absence of certain potentially important variables such as nutrition, health status, and detailed environmental conditions means that these factors are captured only indirectly through breed fixed effects or time trends. The focus on milk yield as the sole outcome does not capture other dimensions of performance such as milk quality, reproductive success, or animal health. These limitations suggest directions for future research that could provide more comprehensive understanding. Future extensions of this research could include expanding the set of breeds studied to encompass a wider range of genetic backgrounds and production systems. Incorporating additional time periods, particularly more recent data, would allow examination of how relationships have evolved over time with continued genetic selection and changing production practices. Including additional explanatory variables related to nutrition, health, environment, and management would allow more detailed understanding of the mechanisms through which breed, age, and lactation period affect milk yield. Examining additional outcome variables would provide a more complete picture of overall performance. In conclusion, This study has employed panel data analysis methods to examine factors affecting annual milk yield in 5 different goat breeds raised in Turkey between 1991 and 1997. The findings indicate that both age and lactation period have positive and statistically significant effects on milk yield. Significant differences were found among breeds in terms of milk yield, with the Cukurova breed showing higher performance and the pure Damascus breed showing lower milk yield. The panel data analysis methodology proved to be an effective approach for analyzing this type of data structure where both cross-sectional variation among breeds and temporal variation over years are important features. The study demonstrates that breed selection and production management play critical roles in productivity in goat breeding operations and shows that panel data analysis can be used as an effective method in animal production research. The careful attention to statistical methodology, diagnostic testing, and appropriate model selection procedures enhances confidence in the validity and reliability of the findings. The results provide useful guidance for producers making breed selection decisions and for researchers conducting similar analyses of livestock production data. The methodological lessons from this study can be applied to other research questions in animal science and agricultural economics where panel data structures are present. Thus concludes this comprehensive lecture on the panel data analysis of factors affecting annual milk yield of different goat breeds. Thank you for your attention, or alternatively, your restful repose during this session of the Boring Lecture Series. We hope this has been a useful resource for your studies or a supportive environment for your rest. This lecture was narrated by Dr. Owen On, an artificial voice for the Boring Lecture Series. Please see the description for further resources and the complete citation to the primary literature discussed throughout this presentation. If you found this level of detail sufficient, please consider subscribing for our weekly sessions. We will now fade to soothing bioacoustic sounds and ambient tones to allow for a period of quiet reflection or continued rest. Good night.